Alright, so here we have an example of a potassium channel. Okay, so it's mentioned here. In the book they don't say what regulates it, right? For those of you who read the book, they're kind of vague on it. It's actually a pH regulated channel, this bacterial protein that they show you in the book. So this is going to be kind of a molecular biology question I'm going to ask you. What kind of experiment can you use to determine which domains are responsible for the cell activity of a channel? Okay? So in other words, we have a channel in the cell. How is this channel going to allow the passage of one molecule and not another molecule? Can anyone give me an example of how a channel, of how a hole can allow someone else to come through? Someone else. I guess I could use a person example. How do you keep some people out of a club, let them into a club? Pardon? Bouncer, right? So we're talking about selective. So how does a biological membrane do something like that? How does a biological membrane let some ions in and not others?
Have the right diameter.
Okay, so two people said the diameter, the size of the opening. So if a pore is too small, it won't allow the passage of certain molecules. Now here's the problem. This is a channel for potassium. What's bigger, potassium or sodium? Potassium is a larger atom than sodium. Alright? So if the diameter is the only contributing factor, how are you going to use a channel's diameter to select for something that's bigger rather than something that's smaller? So it's kind of, yeah. And it's pretty cool how they do that. Obviously, there's a way. We'll come back to this question afterward, but one thing you guys do have to realize is, again, we're trying to make an environment that's favorable to the passage of potassium. So if we look, that was one monomer in the molecule. Here we're seeing two molecules in the monomer, and these purple circles are going to be potassium passing through the channel. So, and this is a top view, obviously, of potassium going through. So, if this is the site that's going to select for the passage, we call this the P domain of the protein. This is what's going to be selecting for the passage of potassium. And as we said, potassium is actually larger than sodium, so how is it selecting through? So we look at these diagrams. I should have prettied this one up. But if we have potassium on the left, sodium on the right, again, as we see, it's larger. So when these ions are in water, that means that the water molecules are actually binding to these atoms, right? So that's what we're seeing here. These are water molecules that are being bound. This larger one is what? This is oxygen or hydrogen? Oxygen, right? So the oxygen is actually binding to the ions of potassium. It's bringing it towards the channel. Okay? Now, it shows 4 water molecules bound. It would actually be something like 8 water molecules. And same thing with sodium. Now, when these water-covered atoms move towards the P channel, does anyone know? There's 2 ends to an amino acid. Okay, so before we talked about how amino acids were used to create hydrogen bonds or whatever. So there are 2 ends. What's one end of an amino acid? Protein. There's the amino terminal, and the other one is the carboxy. Okay, so what does carboxy look like at the end anyway? COOH. What's water? OOH, kind of. So, that sounded like, but it's true. That's how these things pass through. So the OOH, so what happens, if this thing is moving, and imagine my arm is, one of the P domains, and then there's another one here on this side, it's going to be offering up O's, oxygen, to the molecule, such that this potassium ion has to be large enough to reach both of them. Are you following? So to be energetically stable, this thing needs to be bound to 4 different oxygens. So because it's larger, this can now move to the P domain, and it can now reach the oxygen molecules on either side of the channel. Okay? And then it could continue to move on. It could reach another 2 oxygen molecules lower down in the channel. What prevents sodium from passing through is that it's too small. So if this is energetically favorable for sodium, once it reaches the channel, it might be able to reach one side, but not the other. Are you guys following? Okay, so this is not energetically favorable. It's only able to bind to one side of the channel. Yes?
Once it binds, how does it unbind? It attaches? Or it blocks it? What's the system that unbinds it?
What's the system that unbinds it? What is going to be driving it? So everything is in equilibrium, nothing's necessarily static. So it doesn't mean that it binds there and stays. It just means it's more likely to stick around for a while before moving somewhere else. So because throughout this channel we're going to have the same low residues, it's going to bind here for a little while, but the concentration is still lower inside the channel, right? So the tendency is still to move downwards. So as long as the environment in this channel is stable and suitable, it's still going to try and reach its lower concentration by moving down the channel, okay? So you might say that there's kind of two forces. It's affinity for the oxygen molecules in the channel, as well as its tendency to travel down its concentration gradient. So the simple answer is, why is it moving down? It's moving down. It's
I'm talking about the sodium now. The sodium binds to one side. Does that like block the potassium from binding? Is that a problem?
this is so energetically unfavorable, it won't stay here very long, right? It's more energetically favorable in water than in the channel. And I think what was described as something like 1,000-fold. So this is 1,000-fold more likely to bind than the sodium. Okay? So it's not going to stay there very long. Does everyone follow that? So, okay, here's another diagram, the stick and ball diagram, just showing you potassium flowing through there. Again, this is kind of just showing where these molecules are going to interact, right? So you can see where if sodium was smaller, it wouldn't get anywhere near the oxygen domains on either side of the molecule. Okay? And again, one thing they go into quite a bit of detail in the book, well, not quite a bit of detail, but I don't even know why they mentioned it, just showing the movement of the molecule with 8 oxygens down the channel. So as it moves into the channel, it would be binding to 4 oxygen atoms here. There would also be, in case you get confused, there would also be some in the plane. It's supposed to be 8 total, okay? And again, as I described before, it's going to move down this channel and eventually go to a point of lower concentration. Before we move on there, I want to go back to this other question. They do talk about it in the book. What kind of experiments can we use to even start looking? Why would we even start looking at the P domain and what's present in the P domain and how that's important? So probably only people who read the book would know, or maybe there's some super smart geniuses. What kind of experiments can we use to identify the different domains? All right, so if ever you take molecular biology, I don't know, or comparative genomics, we can look at the similarities between this channel and a sodium channel, right? We can look at the nucleotides. So what are nucleotides? A, T, C, G. And we can see where is that similar and where is that different from a sodium channel. So what you would find is that there's a lot of similarity between a sodium channel and a potassium channel if you look at the nucleotide sequence and therefore the amino acid sequence of this domain. Whereas there are some differences in the amino acid sequence and the amino acid sequence and the nucleotide sequence between sodium and potassium channels. Right? Everyone follows that? So that's one way. If you're looking at sequences, so our different genes, if we have a gene for a sodium channel, where does it look different from a potassium channel? Then we kind of hone in. We say, okay, the P domain is where the differences are. Okay, what other experiments could we use? Once we've sort of identified that, we know where to look. Is there anything else we could do? Yes?
Knock out that domain through atom.
That's right. That is a biologist's best friend. Study life by destroying it. You knock things out, you knock genes out, you see what happens. So if we see a gene that's coding for the P domain, biologists ask, well, what if it's just not there? If it's not there, then the channel is actually going to lose its selectivity and it's not going to select for sodium. Okay, another thing we could do is just sort of replace the genes. We can knock it out, or we can replace residues sort of one at a time. These are the kind of molecular experiments they use to sort of hone in or pinpoint which domain of this protein is responsible for selection of.